like to present to you about the way to assist pregnancy, which is also known as assisted reproductive technology. We have 6 types of technologies that can be used to assist pregnancy, which are artificial insemination, in vitro fertilization, gamete intrafallopian transfer, zygote intrafallopian transfer, intracytoplasmic sperm injection, and the usage of drugs. As we can see here, the first step is artificial insemination. It is a medical procedure of injecting semen into uterus or cervix for the purpose of achieving a pregnancy through in vivo fertilization. For example, intrauterine insemination (IUI) is used to increase the number of sperm that reach fallopian tube and to increase the chances of fertilization. It is the best choice of treatment due to the sperm disorder. The example of sperm disorder is low quantity and quality of sperm production of the male, and the male produce a sperm that cannot reach the fallopian tube well. This treatment involves injecting sperm through an arrow catheter into the female's reproductive tract. If male ejaculation still contain few or no live sperm, the couple may decide to choose other than artificial insemination. This is the figure of female reproductive structure. This is the collection of the sperm. This is the ovary, cervix, vagina, catheter, body of uterus, and fallopian tube. This collection of sperm will be injected into the body of uterus by using the catheter, and this sperm will undergo further implantation. The second way to assist pregnancy is by using in vitro fertilization (IVF). It is procedure of obtaining egg and sperm from male and female, which is then place them together in laboratory dish to facilitate fertilization. Fertilized eggs are then transferred several days later into the female's uterus, which is the site of implantation and embryo development, hoping that the chances of normal pregnancy will occur. Basically, in this treatment, there is 4 steps of IVF procedures, which is Step 1, ovulation induction, Step 2, egg retrieval, Step 3, fertilization, and Step 4, embryo transfer and implantation. As for the first step, it is ovulation induction. Fertility specialists will monitor female ovaries and the timing of the egg release. This is to make sure the ovaries are producing eggs and the hormone level are normal. At this time, the female will take fertility medicines or hormones to stimulate the ovaries to produce 1 or more eggs. If female cannot produce any eggs, the doctor will suggesting about donor eggs for this process. For the second step, egg retrieval, a very thin needle is passed through the upper vaginal wall. With the use of vaginal ultrasound, fluid is removed from the follicle under gentle suction. The egg is isolated from the follicular fluid and placed in a culture dish that containing nutrient media, which then transferred into the incubator for 48 hours. The third step is fertilization. During this time, the most active sperm, either from the partner or donor, is mixed with eggs in a special chamber. The sperm is injected into the eggs and are placed in the incubator. The sperm and egg are monitored to make sure a healthy embryo develops. The final step, which is embryo transfer and implantation, the healthiest is the one that will be transferred. A speculum is placed into vagina, and then the embryo are transferred via catheter that is placed through the cervix into uterine cavity. After IVF process is complete, bed rest is often advised for at least 24 hours. We have also provided a video for you guys so that you guys can be more understand about the IVF process.
The menstrual cycle of fertile women usually lasts about a month. During this time, and under the influence of the body's own natural hormones, a tiny sac within the ovary containing an egg grows to about the size of a grape. At a point midway in the cycle, this egg is released from the ovary and ovulation occurs. Ovulation is also regulated by hormones. The egg, which from this moment on is ready for fertilization, then passes into the uterine tubes to await the arrival of fertile sperm. Ovulation is a naturally occurring event, and the desire to have children is a similarly natural instinct. But that instinct cannot always be fulfilled. The causes of unwanted childlessness are numerous, but so are the means of treating them. With the technique of intrauterine insemination, sperm is provided either by the male partner or by a third-party donor whose sperm has been stored in a deep-freeze sperm bank. Before insemination, the sperm samples are prepared in the laboratory. They are assessed for safety, quality and quantity and then passed through a filtering system for grading and washing. With a syringe and fine tube, the prepared sperm cells are then introduced directly into the cavity of the uterus beyond the mouth of the womb and the cervix. They immediately make their own way towards the uterine tube. The objective of IUI is to bring these sperm cells as near as possible to the egg. Nevertheless, only the most mobile sperm cells will actually reach the egg, and only one will penetrate the outer layer of the egg to achieve fertilization. But just as in everyday life, there are many times that fertilization fails and the cycle passes without a pregnancy.
In vitro fertilization may be performed if a woman has been having trouble getting pregnant, a condition known as infertility, or it may be done if a woman wants to have a child without a male partner. Before in vitro fertilization, a woman will receive fertility medication that causes more than one egg to grow and mature in the ovaries. A man will provide a semen sample so that the healthiest sperm can be collected for fertilization. If the male partner is completely infertile, also known as sterile, or the woman doesn't have a male partner, a donor may be arranged to provide sperm for this process. In vitro fertilization consists of three main procedures: follicle aspiration, fertilization, and embryo transfer. During the first procedure, called follicle aspiration, eggs will be harvested from the ovaries. At the beginning of this procedure, an ultrasound probe will be inserted into the woman's vagina so that her doctor can view her ovaries. Inside the ovaries, the doctor will look for follicles. Each follicle is a fluid-filled sac that contains an egg. Then the doctor will insert a long, thin needle into and through the wall of her vagina and guide it to the ovary. A suction device connected to the needle will collect several eggs from inside their follicles. During the second part of in vitro fertilization, called fertilization, the collected eggs will be taken immediately to a laboratory where they will be fertilized. Fertilization may be performed by insemination, where several sperm are mixed with the healthiest eggs. Or the eggs may be fertilized with sperm injected directly into them during a process called intracytoplasmic sperm injection. The fertilized eggs, also called embryos, will be monitored for 3 to 5 days as they begin to grow. At this time, the lab may create a hole in the zona pellucida surrounding some of the embryos. This process, called assisted hatching, will help these embryos implant in the uterus. Some of the embryos will be used right away for embryo transfer, and the rest will be frozen and stored for future use if necessary. The third part of in vitro fertilization is called embryo transfer, which is done 3 to 5 days after fertilization. During embryo transfer, a tool called a speculum will be inserted into the woman's vagina so that the doctor can see her cervix. A syringe will be loaded with fluid containing one or more of the hatched embryos. Then the syringe will be attached to a long, thin tube called a catheter. The doctor will insert the catheter into the vagina, through the cervix, and into the uterus. Once inside the uterus, the doctor will inject the embryos. Then the catheter and speculum will be removed. The woman may continue to lie on her back for about 15 minutes.
The next technique to assist pregnancy is GIF, gamete intrafallopian transfers, and ZIF, zygote intrafallopian transfers. Both of these techniques is procedure used to treat the infertility and to foster the fertilization inside the female bodies. Let's start with the GIF, or gamete intrafallopian transfers. Sperm and eggs are mixed together outside the body in the petri dish before it's being inserted. And with luck, one of the eggs will fertilize inside the fallopian tubes. The successful rate for this technique is about 37% for age 30X and below, and 24% for age 39 and above. The pros for this technique is natural conception and no link to cancer. The cons for the GIF technique is it requires surgery like laparoscopic surgery, and it also may not work because, like I said, with luck, one of the eggs will be fertilized in the fallopian tube, but if no luck, the eggs will not fertilize. It also will become oaths of multiples, that is like conjoined twin, and it's also costing and time-consuming because it requires blood test and ultrasound. The price for this one cycle techniques is about $15,000 and up to $20,000. Now I will tell about ZIF, or zygote intrafallopian transfers. It was when sperm and eggs are fertilized outside female bodies. In the laboratory petri dish, the fertilized eggs are inserted inside the woman's fallopian tube. The pros for this technique is it has fertilized egg, and it has natural conception and no link to cancers. The con for this technique is because this technique is hard to find in Malaysia. The price for these techniques is about $15,000 up to $25,000. The successful rate for this technique is about 22%, depend on the couple fertility problem. If they have a high fertility problem, the successful rate will be coming down. Now I will tell about the procedure for GIF and ZIF. Both of techniques requires ovarian stimulation by the drugs and egg retrieval. For the GIF, they will placing the mixture of sperm and ovum directly into the female fallopian tube. But for the ZIF, they will place the fertilized egg directly into the fallopian tube. The differences between GIF and ZIF: for the first is fertilization takes place inside the female bodies for the GIF, and for the ZIF, fertilization takes place outside the female bodies at the laboratory. The second differences is gamete, egg and sperm are being transferred to the fallopian tube for GIF, and zygote being transferred for the ZIF. The third differences between GIF and ZIF is fertilization may not happen because the eggs and sperm are inserted into the fallopian tube, and for the ZIF, the fertilization are confirmed because it inserts the zygote into the fallopian tube. The process takes about 4 to 6 weeks for both, and for the ZIF, it will best compared to the GIF. Now let's see the differences between ZIF and IVF. For the IVF, the fertilized eggs are inserted into the vagina, which then will allow implantation to occur, while ZIF, the fertilized egg, are inserted into the fallopian tube. The second differences is IVF help in female who have blocked fallopian tube. For further understanding, you can watch the next video.
Sperm injection, known as ICSI. This is the newest form of sperm micromanipulation procedure performed in the laboratory. The ICSI involves the injection of the sperm into the cytoplasm of the egg or the oocyte by using an injection needle, or known as thin glass needle. This method is the most significant development to treat male infertility. The single sperm is injected into the egg cytoplasm. Even testicular or epididymal sperm can be used. This method has a 70% to 100% success rate. This method uses a bypass normal mechanism of the sperm that is used because it uses egg as the interaction to the prior to the fertilization. The pros and cons for ICSI method is: first, the pros. It is a widely practiced procedure. This is because many people around the world use it to achieve pregnancy. Next is high success rate. As I informed earlier, there is 70% to 100% success rate. Next is cons. The first point is complex procedure. This is because it involves the organ inside the body. Next is time consuming. This is time consuming because it takes a longer time. Then it is pricey. As you can see here, the price about $50.
We now continue the another method to assist in pregnancy, which is fertility drugs. Firstly, we have clomiphene. It also known as estrogen blocking drugs. It causes the hypothalamus and pituitary gland to release hormone GnRH, FSH, and LH. This hormone will trigger the ovary to release the egg or ovum. The drug is also effective when used with another fertility method. For your information, there are about 60 to 80% of women who will take this drug when ovulate, and 50% of them will get pregnant. However, there are also side effects for this drug, which is the woman will feel hot flashes, blurred vision, nausea, bloating, and cervical mucus. Second is injected hormone. We have human chorionic gonadotropin, HCG, and follicle stimulating hormone, FSH. HCG is used with other fertility drugs to trigger ovary to release the egg, while FSH is trigger the growth of egg in ovary. Injected hormone have high rate of success in helping ovulation, and 50% of women can pregnant. The side effect of injected hormone is tenderness, infection, and swelling at injection site.